Okay, so what you guys have been going over lately is learning and memory. And this is just kind of a surface level view of things. There's an entire class here that they offer at UTD. If any of you have taken it, it's called the Neurobiology of Learning and Memory. I'm taking it right now. Basically, everything in this section are things that are covered in the course. So if you've taken that class, awesome. This should be a breeze for you. But if not, it's pretty it's not too difficult, I don't think. If you're struggling with any of the concepts, feel free to let me know. Um, but hopefully this will be a little bit easier, at least compared to the motor section, which I know can get a little bit tricky. So we'll start out with some definitions. Learning is just the process of acquiring new information, and memory is the ability to store and retrieve that information. So memory isn't just being able to recall events. It also, what falls under that definition are implicit things. Even muscle memory falls under the definition of memory, and it's something that's been studied for, since the ancient Greeks. But we'll start in the 1900s with Carl Lashley. He made lesions on the cortex to search for the site of memories. This is also called the engram. I didn't see that word in your slides, but just so you know, that's what it's called. He proposed the law of mass action, which basically says that the severity of memory impairment depends on the size of the lesion. So he found that the more of the cortex that he lesioned, the more that memory was impaired. So he thought that there was an equal probability of destroying a memory by lesioning any location on the cortex. So he didn't think that, based off of the findings of his experiment, that there was just one site of memories. And as we go through the PowerPoint, you'll see that the hippocampus is especially important, and he didn't actually lesion below the level of the cortex. So that's why he proposed the theory of the law of mass action. So one of the famous patients that you've probably talked about in your courses before is patient HM. And Scoville and Milner published a famous paper on patient HM who suffered from epilepsy, and they decided to do a bilateral hippocampal lesions to treat his epilepsy. And as far as I'm aware, it did make the seizures go away, or at least lessen the severity of them, but the surgery caused severe anterograde amnesia, which meant that he can't form any new memories. So he had a little bit of a working memory. He could hold a conversation, but as soon as his attention was drawn to something else, or as soon as he was distracted, he lost recollection of what was going on. So if he met a nurse, they talked for a while and then the nurse left and came back, he couldn't remember them. But older and more long-term memories were still intact. So he remembered where he grew up, he remembered his family members and things like that. So the type of amnesia that he called is anterograde amnesia, which means that you can't form new memories. But there's another type of amnesia called retrograde amnesia, which is the loss of memories formed before the onset of amnesia. So if someone does forget their family members or where they live, but they're able to form new memories, then that would be retrograde amnesia. There's one way that we study memory, especially in rodents, is using spatial memory. And we know that the hippocampus is important for spatial memory. So rats are actually really smart when they do this Morris water maze. Often they're called extra maze cues, are placed around the room so that the rats can orient themselves in this large water trough. And the water is just a little bit cold so that they want to find the platform. And so in this specific task, the platform is in the same place each time so that the rats can use those extra maze cues to find the hidden platform, which is just beneath the surface of the water. However, rats with hippocampal lesions take longer to find this platform, and they don't use those extra maze cues to learn where the platform is as easily. So one of the best ways to study memory, although it can be detrimental in humans, as seen with HM, is to make a lesion to an area and see what an animal can't or cannot, or can or can't do with that lesion. So this slide got a little bit messed up, but that's okay. So the working model of memory is that we intake sensory cues, and they can be visual, auditory, any kind of sensory cue. Then it goes into what's called your sensory register, and that's kind of the echo that you have. So if you've ever heard someone and not understood what they said the first time, but a couple of seconds later you realize what they said, that's your sensory register. And then the next place, imaginary place that it goes, is into short-term memory, which lasts about 30 seconds. And an offshoot of short-term memory is what we call your working memory. So that's if you've retrieved a memory and you're thinking about it, or you're thinking about a certain smell or a certain person, that would be in your working memory. So then when you go between your short and your long-term memory, that's either called rehearsal and consolidation. So going from short to long is rehearsal and consolidation. So when you're trying to memorize something, you'll rehearse it. If you've ever had to memorize a speech, that's the best example of rehearsal and consolidation. With the more practice, the more likely something is to be stored in your long-term memory. And then when you pull something out of your long-term memory into your short-term memory and into your working memory, that's called retrieval. So we define memory a couple different types of ways. So the 3 stages of memory are encoding, consolidation, and retrieval, and those are also right here as well. So encoding would be sensory cues to sensory register to short-term memory. Consolidation is short-term memory to long-term memory, and long-term memory to short-term memory is then retrieval. And the 2 types of memory that you'll often hear about are declarative and non-declarative memory. So declarative memory encompasses anything that's conscious. This includes history, words, just what did you eat for breakfast today, things like that. And then under the umbrella of declarative memory, there's 2 more kinds of memory, which are episodic and semantic. So episodic memories are personal, things that happen to you in your life, who your family members are, things like that. But then semantic memory are facts and knowledge, like state capitals or who's the president. And then non-declarative memory encompasses motor skills, associations, priming cues, and problem-solving skills. And these are often non-conscious. So we'll talk about a couple types of non-declarative memory later in the PowerPoint. One of them is classical conditioning. It's not something that you're aware of. It's not something generally that you can consciously control. So some examples of non-declarative memory are skill learning, priming, and conditioning. So going back to patient HM, he could not form new declarative memories, so he couldn't form any new conscious memories, but he could form implicit non-declarative memories. So the example that's presented to you is that he was taught a task in which he had to look at his hand in a mirror, and he had a pencil in his hand, and he had to trace a shape only by looking in the mirror. And he was actually able to improve the speed and accuracy at which he could draw the shape, just even though he couldn't remember doing the task. So it's not every type of memory was impaired. So that gives evidence that the hippocampus may not be the primary location for non-declarative memories. Another case study that you guys learned about was patient KC. He had severe anterograde and retrograde amnesia, which means that I believe he had a motorcycle accident. So he had amnesia from before the accident, and he couldn't form new memories after the accident. But the amnesia was only for episodic memories, and his semantic memory was intact. So he couldn't remember personal memories, but he could remember words and math and historical facts, which is quite interesting. So again, this suggests that the hippocampus is specific for episodic memory because they did know that he sustained damage to the hippocampus. So aplesia are also known as little sea slugs, and they are studied extensively because they have a very simple nervous system, but also their nerves are large and visible to the naked eye. And they exhibit non-associative memory involving just one stimulus. So Eric Kandel studied aplesia, and he won a Nobel Prize for his study. And he basically discovered habituation and sensitization, and we'll focus on habituation. So on the back of the animal, they have a little gill and they have a siphon. So normally, if you touch the siphon, I think Kandel did it with a paintbrush, or if you squirt water on it, then their gill retracts, and it's just a reflex. But if you repeatedly stimulate the siphon, then the animal doesn't retract his gill as much. Now, this isn't something, these are very simple animals. It's not something conscious. It's just called habituation. And then you can actually reverse the process through sensitization, which is if you pair stimulation of the siphon with shock on the tail, then they will actually start retracting their gill again. So these are very, very simple kinds of learning, but they serve as a great foundation for talking about more complex forms of memory. Classical conditioning is an example that's used a lot. It's pretty simple to understand, but defining your terms will be really key for this. From when I took the course, I don't remember exactly if Dr. Raboune asked questions like, what's the unconditioned stimulus, what's the unconditioned response, and the conditioned stimulus and the conditioned response. But especially for taking future classes, I think those are some really important terms to know. So I'll walk you through that. Classical conditioning was, Pavlov inadvertently studied it. I believe this wasn't what he was intending to discover, but as a great scientist, he realized that something he didn't anticipate was happening, so he looked into it more closely. So he would ring a bell before bringing dogs food, and he noticed that each time he brought them the food that they would salivate. So without the bell, food would cause salivation. So that's why food is the unconditioned stimulus. Any dog in any environment will salivate probably when food is brought near them. However, the bell is paired with the food, so the bell does not elicit a response on its own. So before pairing, it's called the neutral stimulus because the dog isn't going to salivate to the sound of a bell unless it's being paired with something. But then during conditioning, the bell is associated with the food and it causes an unconditioned response. And with enough repetition, the bell will become the conditioned stimulus because you had to condition the animal to have a response to the bell. And then salivation in response to the bell is called the conditioned response. Does that all make sense? So salivation can be unconditioned or conditioned depending on what it's in response to. It's important to distinguish classical conditioning from operant conditioning, which I'll talk about in the next slide, because classical conditioning is between a stimulus and a stimulus. So in this case, it's the food and the bell. But operant conditioning is between a stimulus and a response. So operant conditioning is also called instrumental conditioning. It involves reinforcement and punishment. And I think the trickiest thing about this is I'm not sure if she went into positive and negative reinforcement and punishment, but it's an important part of operant conditioning. So positive or negative does not refer to whether it's a good thing or a bad thing. Positive and negative refers to if you add something or if you take it away. So one really good example that encompasses all four of these options is drug use. So reinforcement makes a behavior occur more often, and punishment makes a behavior occur less often. And positive is when you add something, and negative is when you take something away. So positive reinforcement, I'll use the drug use example. So when you take a drug, then it induces a euphoria, and that increases the probability of taking more drugs. So it's reinforcement because you're more likely to take the drugs after you take it the first time, and it's positive because you're adding a euphoric state or taking away pain or something like that. But then negative reinforcement would be something like I suppose what I said before wasn't correct. If you were taking away pain, let's say you take a painkiller, and you take away pain, then that's a negative reinforcement because you're taking something away, but it's perpetuating a behavior. So positive punishment would be, say, an overdose. Something occurs, you're adding something to the situation, which is an overdose or a hospital visit or something like that, and it decreases the frequency of behavior. And negative punishment would be if drug use results in losing your job or losing your friends or something like that. You're taking something away and it decreases the frequency. So I think making a little chart like this is the most helpful way to make these distinctions. So there's a picture in your slides that talks about the pathways for the different kinds of memory. I would be definitely familiar with the different brain regions involved, although it can, a memory is the core of who we are. So there's a lot of brain regions involved, and it wholly depends on the type of memory that you're looking at. So for implicit memory, we're looking at structures like the substantia nigra, the basal ganglia, the ventral thalamus, and the premotor cortex. So if you think of the classical conditioning example, it's a motor response. Salivation is a motor response. or in rats, freezing in response to a fear stimulus is a motor response. So that's why these structures are often involved in implicit memory, whereas the hippocampus is key for declarative memory, especially episodic memory. So now we're going to talk about the underlying cellular mechanisms of memory, but one thing that I want to say right off the bat is that LTP is just a model for memory. And as we start talking about it, you'll see that LTP is induced by high frequency stimulation. So it's not a one-to-one correlation. And LTP is thought to be the underlying basis of memory, but of course, we can't say that for certain. So LTP and memory have a lot of similarities, but they're not exactly the same, and it's important to understand that. So Raboune Cajal was the first to propose that memory is actually stored as anatomical changes involving the strength of neural connections. And that's now called synaptic plasticity, which is the reconstruction of dendritic spines. Donald Hebb proposed that neurons that fire together wire together, which basically means that if a presynaptic and a postsynaptic cell are active at the same time, then their synapse will be strengthened. But neurons that are out of sync will lose their links. So if the presynaptic cell fires and the postsynaptic cell doesn't, then the synapse is likely to be weakened, and that's called long-term depression, whereas neurons that fire together, wire together refers to long-term potentiation. So Bliss and Lomo famously studied LTP, and they also kind of accidentally discovered LTP, which is kind of interesting. They stimulated the perforant path and recorded in the dentate gyrus, and this is in the rat hippocampi. So this, what you see in the picture right here, is called the trisynaptic circuit. So they stimulated, I don't think you can see my mouse, but they stimulated the perforant path and recorded in the dentate gyrus. So first they gave a normal stimulus that wasn't, high frequency or low frequency, and they recorded the response. And then they gave a high frequency stimulus or a tetanus, and then they went back to recording the response from the normal stimulus. So what they found was that giving that high frequency stimulus actually enhanced the response to the normal stimulus the second time it was given. The response was above baseline, and that's what they called long-term potentiation. So I don't actually have a slide about this, but long-term depression is the opposite. So if you gave a normal stimulus, and then you gave a low-frequency stimulus, and then you gave the normal stimulus again, you would expect to see a reduced response to the normal stimulus, and that's called long-term depression. So this is probably the key slide and the key concept to know from the second half of the learning and memory section, because it's the cellular mechanism of hippocampal LTP. So first, let's say we're looking at a glutamatergic neuron. So glutamate activates AMPA receptors, and if you'll remember, AMPA receptors are ionotropic glutamate receptors. So glutamate binds to the AMPA receptors, the AMPA receptors open, and sodium can enter the cell and depolarize the cell. Obviously, this is just a graded potential, so it's not enough to cause an active potential yet, usually. And so as sodium enters the cell, it begins to depolarize, and NMDA receptors are special because they have a magnesium ion block in them. So when the cell is at resting membrane potential, even if glutamate binds to NMDA receptors, they won't open because they have a magnesium ion block. It's often called a coincidence detector because you have to have a little bit of depolarization to kick the magnesium block, and then the NMDA receptors can open. And unlike AMPA receptors, which are only permeable to monovalent cations, NMDA receptors open and actually allow calcium to enter the cell. And calcium is a key second messenger involved in a lot of cellular processes. But in regards to memory, or regards to LTP, the calcium influx triggers a phosphorylation cascade via protein kinases. So at some point, I'd like you guys to maybe type in if she went into more detail than I'm presenting here, or if I guess you would have learned these after the break. And I heard from a student that she's not having live lectures. So I basically copied down what I saw presented in the slides, because these phosphorylation cascades are pretty intricate, but she didn't seem to go into too much detail about them. So I think I would know the bigger picture, but let me know if she said otherwise, please. So targets of these protein kinases are, targets of the calcium and the protein kinases are CAM kinase 2, protein kinase A, which is also called PKA, and protein kinase C, which is PKC. So the short-term effects of LTP is trafficking of AMPA receptors to the membrane. So if you have more AMPA receptors inserted into the membrane, then depolarization is likely to occur more quickly. But this is only a short-term effect. So without protein synthesis, you won't have the long-term effects of LTP. So the long-term effect of LTP is the activation of CREB, and CREB is a transcription promoter that alters gene expression and changes synaptic structure. So earlier when we were talking about synaptic plasticity and talking about the reshaping of dendritic spines, this is CREB, and protein synthesis is responsible for the reshaping of those spines. And LTP also induces a retrograde signal that tells the presynaptic cell to release more neurotransmitter. So there's a lot of processes happening at the same time to enforce LTP, just to make sure that if one mechanism doesn't work or is slow for some reason, then there's other mechanisms working towards the same goal. So a little bit about memory impairment. Age-related memory impairment is totally normal, and it starts occurring actually around your mid-20s. So age-related brain mass declines doesn't actually result from the loss of neurons as much as the loss of neural connections. And as Raboune Hall said earlier, that really is the basis of memory. Is that, it could be those little finite changes and synapses, and that's the most likely, hypothesis for what memory is right now. So this neural connections can be damaged by the loss of dendritic branches, decreased cell size, or the loss of glial cells in the hippocampus, and these are all normal with aging. So there are two things that seem to protect against age-related memory decline, and those are early enriched experience and late enriched experience. So you've probably heard before that, giving children a lot of stimulation when they're growing up is very important because it promotes nerve growth factor expression, it reduces cortisol levels, and it enlarges neural networks. But just as it's important to stay stimulated when you're very young, it's also important when you're very old. So I always hear, read books when you're old and do puzzles and do things that are stimulating, because they actually protect against memory impairment. So that's all of the slides that I have for this, surprisingly. If you have any questions, please let me know. I'm going to stop recording at this point so you guys can speak up and ask questions and not worry about your questions being recorded.